Sir, please start. Sir, kindly proceed, sir.
আচ্ছা আমি
Yes. Habibullah, sir, kindly proceed.
হ্যাঁ হাবিবুল্লাহ স্যার বলছিলাম, শুরু করব। হ্যালো
হ্যাঁ স্যার করুন, হ্যাঁ স্যার করুন।
হ্যালো
হ্যাঁ স্যার করুন স্যার।
আতঙ্কটা ঠিক আছে?
হ্যাঁ স্যার শুনতে পাচ্ছেন, করুন। হ্যাঁ
ওকে। Bajkul Milani Mahavidyalaya, Department of Zoology, with collaboration with IQAC, Bajkul Milani Mahavidyalaya, একটা ইন্টারন্যাশনাল সেমিনার, ওয়ান ডে সেমিনার রিকগনাইজ করেছে, অর্গানাইজ করেছে। এই সেমিনারে বিভিন্ন যে রিসোর্স পার্সন আছেন তাদের বক্তব্য আমরা শুনে নেব। আমাদের ওখানে আছেন চিফ গেস্ট হিসেবে ডক্টর অনিল মহাপাত্র, সায়েন্টিস্ট অ্যান্ড অফিসার ইনচার্জ Zoology Survey of India, Odisha. He there are 50 UCs. He published more than 100 papers in international and national journals, lots of books, chapter, editor of many journals. First of all, the chief guest, Sir Dr. Anil Mahapatra, delegate keynote address. Hello.
Yes sir, yes sir.
স্যার আছেন আমাদের সঙ্গে স্যার অনিল মহাপাত্র, ডক্টর অনিল মহাপাত্র স্যার। হ্যালো।
Anil sir, kindly unmute your mic, sir.
হ্যালো। অনিল স্যার, ডক্টর অনিল মহাপাত্র স্যার।
Sir, kindly call on Anil sir. He can't hear us.
দীপাঞ্জন বাবু আছেন? হ্যালো। নেই। অনিল স্যার আছেন আমাদের সঙ্গে?
হ্যাঁ,
हेलो सर,
कुछ सुनाई नहीं दे रहा है उसका आवाज। तो
सर प्लीज यू रिप्रेजेंट।
तो तुम बोल दो शुरू करने के लिए।
हाँ सर।
अनिल सर, क्या आप सुन रहे हैं सर?
हेलो
सर, अनिल सर सुन रहे हैं क्या? आप सुनाई दे रहा है सर आपको?
nothing. কোনো সমস্যা হ্যাঁ স্যার আপনি শুরু করুন স্যার প্লিজ স্টার্ট
Anil sir, please start. Proceed, sir. We are waiting for your deliberation, sir. Kindly start.
start করছি, okay. Thank you very much. Extremely sorry, nothing was audible to me. I hope I am audible to everybody.
Yes, sir.
Am
Yes.
I audible to all?
Yes. Yes, sir. Yes.
Yes, sir. Yes,
Yes,
sir.
sir. Go ahead.
Am I audible?
Yes, sir.
Yes, sir. Yes. Please go ahead, sir. Please, sir.
I hope there is some technical glitch persisting with this. Let me start. I cannot hear anything from other side, but as per the telephonic call, I am going to start. Respected SS Mishra sir, Professor Prashant Chakravarti, Dipanjan Ray, then the principal of Bajkul Milani Mahavidyalaya, Pijus Kanti Danpat. I had seen some taxonomic genius like Dr. R.P. Burman sir, Dr. Kaur, Mr. Dr. Das. I had seen many people in this forum. First of all, I welcome all of you, and I thank the organizers for this opportunity to interact with many taxonomic related persons. As per the request of Dr. Dipanjan, I have prepared a small lecture on current trends in marine fish species discovery in India. When it comes to taxonomy, it's really a neglected part of science. When we talk about the taxonomy of any kind of animals, it's almost a neglected part. And when as a zoology student, when we talk on taxonomy, it comes to the last of preference probably. Even if when we are students, we used to do like that. So, and when it comes to the taxonomy of marine fish or any type of larger organisms, a small thing comes to our mind that it's overexploited, and maybe most of the research scope is over in this group, and we may or we may not be able to excel in this group of animals in terms of taxonomy or biodiversity. So I'll give you a glimpse of the things that what is happening in India and what can happen in India. So basic species discovery, when we talk about total available species names for fishes is more than 60,000. Total valid species is more than 35,000, and almost 50% belongs to freshwater, and the rest of the 50% is marine. Almost available generic name is 10,000, of which only valid generic name is more than 5,200. Valid generic names for marine fishes is more than 3,000. Very few are exclusively brackish water, means exclusive brackish water fishes are very less as per as on August 2020 data. It's less than equal to 200 only. Then when we come to the fishes of India, as many as 3,632 fish species, which is around 10% of the world record, are recorded from India till date, of which total 216 species under 9 genera, 37 families, 12 orders are cartilaginous, then rest of the things are bony pieces. Freshwater, nearly 850 species are from freshwater, and marine is 2773 species. So this shows that the volume, the number of species in marine is much more and widespread. The thing is, I am talking about widespread or localized. The speciation in freshwater fishes are much more because of the restricted boundaries. But marine species, it can be recorded from Pakistan, it can be recorded from India, it can be recorded from Myanmar, it can be recorded from Bangladesh, like that. Means it is having a wide range. So discovery chances for particular species from a single country becomes almost minimizes to a certain extent. So if we see the description of fish species worldwide, it includes all freshwater and marine species. So you can see it's almost always around 300 to 500, 600 like that every year. But the case varies when we talk about marine. When we go about marine, the data are taken from catalog of fishes. Even if the decadal data you find in 10 years, it's 150. Though last 10 years data is not yet compiled. I hope in last 10 years, lots of marine fishes has come up and it will cross 150, and it will be around 600 marine species in last 10 years. So the thing is, the discovery in marine is comparatively lower than freshwater scenario. Now I am going to give you a glimpse of the top 50 authors who has described marine fish species or fish species worldwide. You will find a very less number of Indians, or I can say within top 50 there is no pure Indian person who is there. They have worked in India, but all are foreigners, and they have worked a lot and described many species. And when describing a new species, it suddenly comes to our mind that if we are going to describe a species, it may be synonymized later on. It happens. But if you see, even if workers like Gunther, Blicker, their average is less than 50% or like that. If you see Randall, it's 96%. It may goes down even if our species, what we are describing today, it may after 30 years, 40 years, some technology will come and it may be synonymized. So if you have sufficient proof that it can be described as a new species, please go ahead with that. There is nothing. If you have data, you have proof, there is nothing bad in describing a new species. But don't describe a new species with just not comparing with many congeners. Then I am trying here to show you some of the great Indian scientists who has described many species in freshwater condition. I had shown you in world the pure Indian scientist whose number comes at 98, Professor Viswanathan. He has described enormous species, and his validity percentage is 91%. So but if you see Indian scenario, even if LK Singh sir is describing nowadays many species, and Dr. Ramadevi from JDSI, they are the genius worker, AGK Menon. But if you see the number of discovery and their place in world taxonomist list, you will find their number at 100 and above 200, 300 like that. So the work in India, though we have 10% of species, the work in India by Indian scientists is very less. Then when it comes to marine species, you can see all young persons. I hope all are now working except Talwar and Ramarao. All are in present working group. I can say Binis, myself, Talwar and Ramarao, then Mishra sir, Achilles, Dipanjan and Swarup. The reason is actually we may have the concept that there is very less opportunity in marine to discover any species. That is the major cause of this scenario present days. So the 100% validity, you can see me, Vinis, Ramarao, Achilles, Dipanjan, Swarup, and these species, we cannot claim that it will be 100% after 30 years, 40 years. It may goes to 90%, 95%, or it may goes to 50%. So it's a process. Taxonomy or describing a species is a process. It's not that if you are describing it should be always correct. We have seen that in many cases that species has been described, then synonymized, then revalidated by us or somebody else. So it's a process. Then when we talk about discovery in India, it's about all taxa. You see, in India, about 300 plus species has been described in recent years every year. But when we go to fishes, you see that 20 average 20 freshwater fishes have been described every year. But if you go to marine, the number comes to a very odd, I can say, very less number, and this might be due to less number of workers, or this might be due to the diversity is more and the exploitation is more, and less number of people are working in this condition. Then I'm coming back to new record to in for country. When it comes to new record for country, you can see that marine species always dominate. The reason is this is the total number of species and marine species. You see that if some species has been described from Japan, Taiwan, or some of the neighboring countries like China or Bangladesh or Vietnam, like that, they have been described in from India and recorded as new record. The thing is, the new record in marine is much more than freshwater, because freshwater, due to geographical restriction, you have all chances of describing new species than new records for a country. Only if some neighboring country species may be there with if some same ecological condition or same nature is there. Then, while working out the type specimens are very much required in taxonomy. Type specimen means I know there are many starter people here, students particularly, they might not be knowing what are the holotypes. Holotypes means the first specimen from which the species has been described. And while describing a congener as new species, you may have to verify these species of from different museums. So museum plays a very key role in describing new species. But if you see India, probably JDSI is having the largest stake, and its number is probably 15th number in world. And we host a good number of type specimens with us. Then I'm going to give you a glimpse of current taxonomic tools, and I hope Mr. Sir will carry forward from here. Current taxonomic tools, when we talk about tools, it comes to me, first tool comes as morphotaxonomy or classical taxonomy. It must be for identification of marine fishes. I feel you may be a taxonomist or a molecular biologist, but you should not neglect this particular part. If you are neglecting this part and directly going to the technology part, it may lead you to a black hole, and you may not be able to guess the right thing or right description part, which we'll be discussing in latter part of my lecture, maybe after 5 minutes. So these are some of the proportionate key characters you need to take care. Maybe fin formula, maybe length, then some proportionate length, then gill racker counting. These are all age-old techniques that I can say Burman sir is there. He was also doing with that, and we are also relying on these technologies. And this is probably the best way to start the taxonomy. And then osteological parameters, if you are going for certain groups like Scorpioniformis or Anguilliformis or many groups of fishes, you need the osteological data set so that you can add some more data or some more features to the morphotaxonomy. So these are particular bones are meant for particular function, and according to the adaptation of the species, those bones are been changing. So it's one of the major character nowadays, particularly in Anguilliformis, which we used to take care of. And I trust me, it's a tool. The osteology or vertebral counting is a additional tool. It's not the pure taxonomy from which you can conclude anything. Then it comes molecular taxonomy. Molecular taxonomy is a blessing, I can say, to the taxonomist, but the blessings should be utilized in a proper way so that you can conclude a better result for this. When we come to this molecular taxonomy part, what happens nowadays, the young generation, what they are doing, they start back calculation. They will do pure molecular gene sequencing, maybe cytochrome C oxidase subunit, and they will match with it gen bank or any available sources, and they will conclude that matching that this is this species. Please don't do that. I can say the reason is, suppose I am identifying some species which is misidentified by me, and you are doing, and I am uploading the gene of that species as a different name, and you are starting with the same species, and you are doing molecular thing and uploading due to my fault from the beginning, while loading that species in the gene bank, you are identifying a faulty sequence of a faulty species to your, and you are naming that unit to a different species. So it creates a havoc and it creates all sort of nuisance. Now I had seen many papers where they have done back calculation, even if new taxonomic description of many species, where they have done taxonomy, but they have never uh, dealt with the original taxonomy. If you are going with that, recently I had seen one eel specimen described from India, and it's around 1.6% difference from its congener by genetics. But when we are seeing verifying that specimen was with me earlier, but we thought it is not a new species, and I hope it will be synonymized soon. So synonym is a process, but it should not be synonymized within a second or within 5 days, 10 days, like that. So this is a blessing. Please use this blessing in proper way so that the taxonomy can flourish. Mm, Then we need both molecular and classical taxonomy for the best approach of taxonomies. And while doing molecular part, there may not be, the same person may not be a taxonomist in morphotaxonomy. It's better to combine, it's better to collaborate. There is no problem with collaborating with other people. You can see the papers with having 10 authors, 5, 6, 7 authors, where 4 people might have gone for collection, identification, morphotaxonomy, and 2, 3 people might have done for DNA analysis and all those things. And so you may have to combine with other countries' counterpart. The reason is, you are bound to combine because we do not have all holotypes with us. You need to compare. You may have to contact Professor Prashant or somebody else, which we used to do regularly. So now also there are many softwares where you can just put the image and identify the species, but it's in a very premature stage. I can say it will be maturing gradually, and the morphotaxonomy will be placed in a different level once these things will come up. If you are loading many photographs to those software companies, they may come up with good data analysis. These are also same type of software. You need to just mark some of the parts of your specimen and upload it. It may give you, if you are uploading 4, 5, 6, 10 species at a time, and that can show you the clades, that there are might be 4 species in this all 10 figures, or there might be 5 species or 2 species in the same clade, though it looks very similar to you, and the software automatically separates them like the DNA clade we are doing. So last 7 years we have some discoveries. I am not going much to that. We have done both morphotaxonomy and molecular taxonomy for this particular species, and if you see, this is named in the name of respected Mishra sir, and this particular species we have encountered in Bay of Bengal, West Bengal coast, and you can see David Smith. We have combined with him because we do not have any congeners of this species with us in India. This is just a short brown one pattern species, and most of these specimens holotypes were in abroad museums, and Smith had verified that. That's why more often we need a counterpart from other country who can have the data for verification of holotypes, and who is having a museum like that. So when particularly eels, we used to take some of the characters like core character, dorsal fin origin, and dental character. Dental character because every species has a different habitat and their feeding habit and capturing of prey style is different. So dental character plays a very crucial role in eels, and vertebrae also plays a very crucial role in that, depending on their adaptation priorities. So these are some of the species which we have described recently. Not going much to this. And there are some new records to India also by our group. Then let us come to the major part. Fish diversity is a declining phase. It's all right. I agree with that. That literally, if you go to any of the landing center, you will see the amount of trashes. Now, government had banned the trashes to bring back to the shore, but literally our trawlers are sweeping the seabed. So definitely the disappearance of some species will be there. The stock assessment result also shows that the declining of many species number, particularly the amount of catch in previous years, 10 years, 15 years back, is not there now. So it's a process of over exploitation, and also some natural characters like climate change and other anthropogenic impacts in seas, habitat loss, pollution, over exploitation. All this affects the diversity, and unlike other animals, it's also applied to the species diversity. Now, need of the hour, the basics when we talk about fish taxonomy is the taxonomist. I can say there are a few taxonomists in the panel who can easily justify that the number of taxonomists now in marine fishes in India are very less. You can count their numbers. Finis, Achilles, I can count. Burman sir, then SS Mishra sir, myself, maybe 3, 4 or others, somebody from CIFE and or somebody from ICAR. That's all. But you will find a good number of people in freshwater groups. And in marine, when we talk about specimen collection, it's a difficult task. More often, we used to depend on the fisherman, and if you have a good connection with fisherman, you can only collect much specimens, and Dipanjan can better explain these things. And the second thing, in India, we most of the organizations want to have their own research vessels where they can go on and trap the fishes from marine as per their wish. So we are deprived of that particular portion. I don't think, except being hiring a trawler and going back to sea, or being hiring a boat and going back to estuaries. That much only we used to do for specimen collection. And the second thing is going to ship. It's a difficult task for many present day students. It's adventurous, all right, but while thinking of adventure, everybody get excited, but during work, after 2, 3 cruises or after 2, 3 trips, they get fed up. It's from my personal experience. I had gone many cruises, and it happens after every cruise. You may be thinking that yes, it's enough. We may not go for another cruise. And if you are particularly going for a summer cruise, it's a very difficult and tough task in Bay of Bengal coast. Then access of type specimens. As I have shown you in the museum types, India is having only 300 to 400 type specimens with us, of which nearly 200 maybe from marine. So for describing a species, you may have to compare with 30, 40 species of species, and you need to compare all the type specimens. So you may have to visit several museums. So you need fund for visiting museum, which is a constraint in India. But still, then for sorting out that problem, you need to collaborate with many organizations or many collaborators, so that you can avail the facility even if without going to the place. And now, what I observed during my work, many people are generous. And they used to provide you data. Even they told that we are not in a position to collaborate for the work. We are helping you for this sort of holotype verification and all these things. Even they used to generously send you the pictures of the species, and you can start after that. So there is a positive thing in this. Then found for survey in different habitats. Actually, if you see, the deep sea fauna of India is almost untouched, I can say. Particularly fishes, very less work has been done in India, and so that I can say in abroad also not much work has been done in deep sea fauna due to constant surveying. Then coordination, collaboration between institutes, which I had already told that it's very much essential for the work. Application of modern taxonomic tools. You need to apply the tools with pride, but please be conscious while applying the tools, and do the basic things, then apply the tools. Always it's advisable. Then identification of gap areas in marine. People used to think that most of these species have been identified. I can say we have started with Anguilliformis few years back, and we have described so many species of Anguilliformis. And I can say you, if somebody tries to now to catch some govids or shark and rays, any group you will find many specimens. But please go to the field. Don't rely only on the market-based survey, and you may not able to land anywhere. So I came to the end of my lecture. From here onwards, Mr. Sir will carry forward about identification. I thank Honorable Director of Zoological Survey of India, S.S. Mishra Sir, Dr. Basudev Tripathi, and Dr. Vinis, my colleagues, and Dr. Todu, Dr. Dipanjan Ray, Swarup, Samitra. They are all, most of them are part of our study. So we work like a group. And thank you. Thank you very much.
Thank you so much, sir, Dr. Anil for his valuable and insightful speech. Now, our next speaker, first speaker, Mr. Subhendu Shekhar Mishra, scientist, and officer, Marine Fish Section, Zoological Survey of India, Kolkata. He discovered more than 20 species of fish, published more than 20 papers in international and national journals, many books, chapters, editors and reviewer of many journals.
Actually, your voice is totally not audible to me. So I am not able to. You can type if anything is there. Please type, and nothing is audible. Your voice is not at all audible.
So I repeat again. Thank you so much, Sir Dr. Anil Mahapatra. Sir, এখন শোনা যাচ্ছে? Hello, hello. Thank you for his valuable and inspirable speech. Now our first speaker, Mr. Subhendu Shekhar Mishra, scientist and officer in charge, Marine Fish Section, Zoological Survey of India, Kolkata. Discovered more than 20 species of fish, published more than 20 papers in international and national journals, many books, chapters, edits and reviewer of many journals. So now our sir Subhendu S.S. Mishra sir, now deliver your valuable speech. Sir achen S.S. Mishra sir. Hello.
Mr. Sir, kindly. Yes, sir.
Hello?
Mr. Sir, kindly deliberate your speech.
Can I start?
Yes, you can start. স্যার শুরু করুন।
I think, sir, you can call Subhendu sir, sir. I think,
হ্যাঁ, শুভেন্দু শেখর মিশ্র স্যার। স্যার শুরু করুন।
sir, kindly make a call to Subhendu sir, sir. I think, sir, cannot hear anything.
Yes, I can hear. Can I start?
Yes, sir. Start. Yes, sir.
Okay.
You can start.
Hello.
Yes.
Yes, sir. You can start now, sir.
Good evening. I think the screen should be visible first. Is it visible?
No, sir.
Only Dr. Mishra's face is coming.
The screen is visible?
No,
Hello.
Hello, sir. No.
No, sir. Not
Is the screen visible?
Hello. Hello.
Hello.
Hello,
Am I audible?
sir.
First of all,
You are audible, but
Audible, but not
screen is not visible, sir.
screen is not visible. So why? Okay.
No, sir.
Okay. I hope it is visible. It is coming
No,
up.
sir. Sir, is not-
Just
Sir, screen visible, sir. Okay, sir. Okay.
Okay.
Oh, okay.
Good evening, everybody. Dr. Anil has already given a detail on his taxonomy, and he has also talked about the diversity and distribution of the pieces. Still, I'm just touching every line and giving only the basic, not going too deep into it. We have seen fishes everywhere in the markets, landing centers, and in our home also, as in aquariums. What fishes are? Fishes are cold-blooded animals. They are having vertebra and gills for the respiration and fins for movement. These three things must be present in a fish, what the shape it may be. They occur in all aquatic bodies, even in polluted water also. Freshwater, brackish water, marine and polluted water, even polluted water. We can find fishes 5 kilometers above the sea level and even 11 kilometers deep in the trench in the sea. They are found minus 2 degrees Celsius below the ice sheets of Antarctica. These are the dragonfish found in Antarctica. And even at 40 degrees Celsius near the thermal vents and hot springs. The fishes, those are evolved nearly 450 million years ago in the Ordovician period of the Paleozoic era. But most of the fishes, you can see, this is the place where the modern fishes start. They are at 400 to 350 million ago in the Devonian period. The size of the fish If the smallest piece is less than 8 mm, you can see the fingertip, the mature adult is present now, and here is the largest fish recorded typhus, which goes up to 20 meters long. Diversity, when we talk about the shape of the fish, they are usually spindle shaped, respondents, cyprinids, sardines. They are all spindle shaped and well shaped to swim swiftly in the water. However, some pieces are compressed on the sideways. Some are with very elongated body, which are we call mostly the eels or spiny eels. It looks like snakes, but as they are having the gills and fins and the vertebral column, they should be called lizard fishes. Here are depressed fishes. Some are very peculiar shape and size, well, middle fish with a long snout and a half beak, and even the flying fish, which usually glide above the sea water. And some are having flute-like mouths, which are called ciguatera. Mostly we divide fishes as either food fishes or non-food fishes. The food fishes, we know it very well: ilsa, recti, pomfrets, mullets, cyanids. They are very common food fishes. And in freshwater, katla, rohu, pangas, tilapia, and the small fishes like salmostoma, puntias, maestas. They are very common freshwater food fishes. And these are mainly most common ornamental marine fishes. And there are several ornamental freshwater fishes are available. In trade, we have about 500 species available in the market as ornamental fishes. Then those ornamental and food fishes, apart from that, others are mostly we call non-commercial fishes. Those are usually small fishes, or even if a large one, it is quickly perishing, or poisonous fishes. Fishes with insufficient flesh, they are mostly bony in content. Those fishes are damaged. Those are considered as non-commercial fishes. But they are usually used for preparation of fish meal, poultry feed, and manure, etc. When I talk to you as poisoners, I can differentiate it into two groups: poisonous and venomous species. Poisonous fish, they produce strong toxin, but they do not bite, sting, or stab to deliver the toxin. Instead, being poisonous to eat, as digestive system cannot destroy the poison. When we eat those pieces, the toxins in those pieces will not be digested in our digestive system, and it will cause poisoning effect. Some of them are known as ciguatera poisoning. I will come to the next slide. And the venomous species are the species which produce strong mixture of toxins harmful to humans, which they deliberately deliver by means of bite, sting, or stab. Venomous species do not necessarily cause poisoning if they are eaten. So these venomous species we can well eat, and their venom can be digested by our digestive system. So here is the picture of at least 4 venomous species. The top right one is the most venomous known fish, is a rip stonefish. If a diver steps on it, it stabs its spine, it may kill the human also. And the lion piece, which is so decorative, is also poisonous, venomous, because its spines contain venom. And the lower one, you can see the stingray. And here are the stings. If you can recall, Steve Irwin, who is known as the crocodile man, was killed by this stingray because one of these sting entered into his left chest, puncturing his heart. So before he was treated properly, he was killed due to bleeding. Some blennies, they also can bite and inject venom. Some of the, you can just recall the Plotosaurus or Clarius, the Madur, they are also venomous. Singi, Heterobusis, Facilis, they are also venomous in that way. But the poisonous, you see this side, left side of the screen is with some tetraodontiformis species. The tetradotoxin they are producing is a highly venomous, a highly poisonous, which is cause stomach upset and kill the man. And some of the eels is also poisonous. They can by eating these eels also cause poisonous effect. And there is the luganics. They can also produce ciguatera toxins. These are some pieces, better pieces, which contain ciguatera toxins, causing ciguatera poisoning. So this includes some pharyngites, acanthorates, even scarites, some epidecalites, luganites, and even barracudas. Normally, from the occurrence point of view, we can distinguish them as primary freshwater fish. Those entirely live in freshwater. Some are secondary freshwater fishes that are usually coastal fishes that can survive in and enter freshwater but common in coastal waters. Then comes the third category, the marine fishes that mostly spend their life in sea but may enter estuaries also. These second two categories commonly called marine estuarine fishes. General classification, from the general view, we distinguish them as cartilaginous species because their skeleton is made of cartilage, and others are bony species. Their skeleton is made of bone. But normally, fish taxonomy is a system of arranged classification of the fishes on fish taxonomy. Is the system of arrangement of animals into groups having common characteristics that express evolutionary relationships. Why we need classification? To organize species into groups and discuss them, identify new organisms, show relationship between organisms. So what is the importance of taxonomy? To identify the animals and their status to be studied towards conservation of the species. The taxonomic studies usually form the base of research and studies. They can be studied for use in medicine, for identification of the natural enemies from fisheries point of view, and for conservation. Taxonomy is the theory and practice of classifying organisms. Study of classification, identification, nomenclature, and phonetic include the entire taxonomy. Classification is the arrangement of animals into groups having common characteristics that express evolutionary relationship that I told earlier also. Identification of determination, the identifying animals by the recognition of certain characters. Most of the characters were just previously discussed by Dr. Anil Mahavidyalaya. The nomenclature, naming of the organism according to the International Code of Zoological Nomenclature. Then phonistics, inventory of native or naturalized animals of an area, that is called fauna. When the taxonomy is concerned, the early systems started from the 308 around the 300 in between 380 BC to 322 BC by Aristotle, who classified the life into two groups, regnum vegetable and regnum animalia. But it took more than 1800 years for the new set of classification to come up. Is Conrad von Deschner in around 1550 brought to hand descriptions and specimens of many novel forms of animal life in parts of the New World. It is Carl Linnaeus, who first classified the animal kingdom into 6 classes: Animalia, Mammalia, apes, Amphibia, Pisces, Insecta, and then beyond that all others are he considered as a bad beasts. Linnaeus is the first to introduce the binomial system of nomenclature, what is now existing and we are following. The common hierarchical ranks, the kingdom, class, phylum, class, order, family, genus, and species. Carl Linnaeus, when you just I am shifting to the Indian scenario, in 1758, Carl Linnaeus published his Systema Naturae, where he included 75 species as from India, but many of them are from Indian Ocean, not from India exactly. From the Indian subcontinent, the first work probably by Patrick Rochelle in 1803, who described 200 species from Visakhapatnam, from the Coromandel coast. However, all these species he described are not in the form of binomial. Hence, his work is not reflected in the scientific nomenclature of the species, but many of his species were later described by other scientists. And the freshwater species concerned, it is Francis Hamilton in 1822 described 260 new species, out of which 177 species are valid on date. Later, there are so many contributors in between also, but I am coming the major work, what is still we consider is the Bible of Indian ecology, that is of Sir Francis Daye's work, The Fishes of India, that published in 1975, 278, 4 parts, then a supplementary in 1988. Then he later consolidated the entire work as The Fishes of British India, where he included only 18 species, 1,418 species under 342 genera. That includes both freshwater and marine fishes from Indian subcontinent. Another worker, the almost the end of the century, who contributed to the deep sea fish study in India, are Alfred William Alcock. In his two books, Illustrations of Zoology and Indian Deep Sea Fishes of Indian Museum, the collections of Alcock was kept in Indian Museum. That both are investigator collections. He participated in RIMS investigator and collected the deep sea specimens from Bay of Bengal as well as from Arabian Sea, and he described 169 species from this area. The major contributors, of course, Dr. Mahapatra has given details. I'm not going to that side, but Blicker, Valencius, Jordan, Gunther, Kohler, Flute, and Snyder, they usually contributed mostly for the study of Indian fish fauna. When we're coming to marine fish diversity, the marine fishes are mostly abundant in coral reef areas. You can see the coral reef here at Andaman, Gulf of Mannar, Gulf of Kutch, Lakshadweeps, and to some extent in Sindhudurg. That is reflected in the fauna. You can see the Andaman Nicobar. It is usually closer to the center of the evolution. The coastal fishes in the coral triangle in the Central West Pacific. Andaman is the meeting point for Western Pacific and Indian Ocean. Andaman water harbors more Western Pacific fishes and more diverse. Total species recorded from Andaman is now 1601. Of course, more than that, after this number I am giving, few more species has already been reported as new records by Dr. Dinesh and others. From 15 more are added up. From Lakshadweep Islands, the faunal composition of Minica Island is more allied to the fauna of Maldives, but not of that of Indian coasts. More than 831 species of fishes recorded from Lakshadweeps. Number of marine fishes known from different maritime states. Each maritime state with the within bracket I have given the length of the coastline. Sorry. Andaman having 1900 kilometers coastline and harbors 1601 species. West Bengal 577 species, and the coastline is just 158 kilometers. Orissa 208 kilometers and 642 species reported. Andhra Pradesh having 974 kilometers long coastline, 783 species, 93 species. Tamil Nadu 1178 species, Puducherry 314, Kerala 745 species reported, Karnataka 562, Goa 168 species. Is a small area, 100-kilometer area, but a good number of species is reported from that area. Maharashtra is reported with 646 species. Gujarat having a long coastline, but only 447 species are reported. Lakshadweep has only 132-kilometer coastline, but reported with 831 species. When it comes to different states, the freshwater fishes, Andhra Pradesh with 158 species, Arunachal Pradesh 213 species. Of course, I have given this 2009 figure, but later on 2016 it is increased to 259 species. Like that, these are reports I have given, corresponding reports, but later on many species has been reported from that area. So it is a minimum number I can say. More than this you can get the species. Bihar, Jharkhand 134, Assam 187, Gujarat 113, Jammu Kashmir, actual report yet to come. Kerala having 188 species. And just from this figure, you can see Arunachal Pradesh 259 is the highest number of species, most possibly, because the northeast area is, you can say, northeast hotspot. We have 4 hotspots we talk about: the northeast region, the Western Ghat region, where the Karnataka and Kerala comes in that Karnataka, Kerala, Tamil Nadu comes in that Western Ghat region. Combinedly, the number is much more. So Maharashtra, including Maharashtra, these 4 states, the Western Ghat is spread over 4 states. So if you will see combined things, maybe it is much more. But otherwise, Arunachal Pradesh, Assam, they are having more species. And in West Bengal, we have only nearly 240 species known from West Bengal. Dr. Mahapatra has already told about the total number of species. It is, but pieces is nearly 1/2 of the recognized extant vertebrates in the world. There are 35,613 species known to date. They are placed in 77 orders, 603 families, and 5,212 genera, of which 17,917 species are freshwater forms. Nearly 50% is marine. It is slightly, freshwater is slightly more than 50, and marine is slightly less than 50. And only few are brackish water forms. The total diversity, I may be differing from that of the figure given by Dr. Mahapatra. I cannot say what is the difference, but my estimation is now it is 3,702 species known from India. This is just 10.4% of the world fish diversity. Freshwater fishes are 992 species, that is 26.8%, and marine fishes are 2,710 species, there are 73.2 species, of which cartilaginous fishes are 302 species, and bony fishes are 2,507 species. The primary species, freshwater fishes, are contributed by just a few orders: Osteoclosiformes, Sapriniformes, Karashiformes, Cilidiformes, Saprinodontiformes, Synbranchiformes, Anamantiformes, Harmoniformes. Of course, Siciliformes also comes here. There, I missed out while I wrote it below. The few pieces of Tupidae, Mugilidae, Sicilidae, Ambashidae, and Cyanidae also include as a freshwater pieces. So these 1, 2, 3, 4, 5, 6, 7, 8 orders, they constitute 969 species, and these additional species 23. The total it comes 992. The marine fishes, they are represented by class Bixini, just in one species, reported by Venomous 2013. But later, Benham et al. They reported an unnamed species of Aptopterus from Kerala coast. They are not sure whether it is, they are sure that it may not be Heptopterus, but maybe some other species. Let us wait for a conclusive study on this species. But anyhow, one Bixini is available in the deep waters of Kerala. The Elasma branchies, there are 11 orders, 39 families, and 198 species of Elasma branch. are available along Indian coast. There are only 2 families and 4 species of holocephali, the chimeras. They are found along Indian coast. I have seen commonly the Nagapattinam area. The class Actinopteri was reprinted by this almost 36 orders. There is nothing new in the naming them, but earlier this Berishi Farmis, Tachyctis Farmis, Holocentric Farmis, they were considered as one group, now separated into 3 orders. Stomti Farmis, Kurti Farmis, Bony Farmis, Karanji Farmis, Akanthuri Farmis, Santrachi Farmis, Acropomati Farmis. These few orders now newly inherited from Perciformes. They are separated from Perciformes. Earlier known as Pleronectiformes, now placed in Karangiformes, and Scopeniformes, they are now placed under Perciformes. What exactly we need? Dr. Mahapatra is also discussed the same thing, but experts on the field of taxonomy are very less. Hence, we need to develop manpower and taxonomic study on fishes. Few workers are available for freshwater fish study, undoubtedly, while the marine study and number are very, very limited. Biodiversity studies done by comparing photographs in many cases, they are many times go wrong. Just comparing photographs and identifying is not at all a proper way. So it is for them I can simply call the desktop scientists. They need to study the piece in the classical way of study, when using other tools that described by Dr. Mahapatra should be used, including the molecular study. But this molecular study sometimes gives problem if it is not used judiciously. Why I need the classical study actually? Here is, I'm giving just one example. A brief comparative account of axial skeleton of 6 polynemes from Bombay waters. They described, they used the Polynemus heptadactylus is used from some species, whereas Polynemus heptadactylus, what is now known as Philemonus heptadactylus, is not occurring in Indian coast. Either it was Polydactylus Milani or Philemonus similis. Most possibly, from the osteological studies they made, we found it is mostly goes with Polydactylus Milani, but not even Philemonus. It is not a Philemonus species even. So proper identification is highly essential when we are going to the biological studies or other studies related to particular species. We should not make mistake in the species names. Here, it is an example of using photograph for identification. The piece on the top is Plectorhynchus ray, having 14 dorsal fin spines. Spines of dorsal fin is 14 in number. In this hemulids, the spines and rays are mostly constant in the species, excepting few aberrations. But the species was identified as Plectorhynchus gutturinus, which is having only 13 spines, and you can very well see the bending pattern. It's not exactly 6 lines, but here in Gutturinus juvenile, it is 3 pairs of dark bands. Here only 5 bands you can see, and even usually in Gutturinus at the size of 10 centimeter length, these bands started disintegrating. But even the specimens of more than 10 centimeter length, These are very prominent bands you can see in Latrinus, in Petroleucus ray. Here, in all Indian literature, there is a species you can find as a Secutor ruconius. This is the original figure by Hamilton as Leognathus ruconius. Leognathus ruconius, it looks, the teeth pattern, mouth pattern is of a Leognathus species. It cannot be a Secutor. What we call a Secutor is now is a Daveximentum, and I hope this is the species actually described as Secutor ruconius, not this one. The second, the B species is described as Secutor ruconius. So it must be considered as a Daveximentum interruptum, not Secutor ruconius. Ruconius is a Leognathus species, not Daveximentum species. The further scientific studies reveals you have to take, you have to keep contact with the what's done all over the world. Earlier, we are considering this species as Trachinocephalus myops, but further molecular studies confirm that Trachinocephalus myops is confined to the Atlantic region, while the Indian Ocean species should be called as Trachinocephalus trachinus. Similarly, earlier we discussed presence of Cryus batrachus in India. However, the Indian species is now known as Cryus margot, not batrachus. Batrachus is now a Javanian species. The Chana orientalis, still in some of the papers I am finding that Chana orientalis is being reported from India. But one must keep in mind, Chana orientalis do not have the pelvic fins, and what the species people use to describe as Chana orientalis, mostly following the identification by Talwar and Jingran 1991, which included at least 4 species under Chana orientalis, and the Indian species are mostly Chana gachua. Orientalis is only endemic to Sri Lanka. It is not available anywhere else. Recently, the most common pomfret we are knowing Pampus argenteus in Indian waters. However, it is now proved argenteus is confined to Western Pacific, and the Indian Ocean specimens are Pampus candidus. You can refer Radhakrishnan et al. 2019 paper to find out when they reselected Pampus candidus and proved that none of the species found in Indian Ocean are Pampus argenteus. In a recent study, we found the Oxydatus dentatus is also a Western Pacific species, and the Indian Ocean specimens, what earlier identified as Oxydatus dentatus, should be known as Oxydatus naxipinus. Yes, I was talking about molecular study. There is some problem in molecular study that Mahapatra has discussed how he was able to get some species identified through molecular studies, and that is true. Molecular studies, when integrated with classical taxonomy, is giving very good results. But here is one of my experience. When I tried to find out the sequence available for Mughal cephalus in bold, I found bold or NCA. I found there is 7 sequences available from India, but 2 are quite different from the other 5. So I was very much sure all the 7 are not Mughal cephalus. Either 2 are Mughal cephalus or the other 5 are Mughal cephalus. Then I checked for Mughal cephalus from Indian Ocean. Obviously, I found the Mughal from Indian Ocean region matches with the two sequences submitted from India, and the 5 other sequences are totally different from Mughal. Possibly, it is a sequence of some other genus, not even Mughal. So, if somebody tries to take the sequence first and match the sequence in the gene bank available, then they will certainly be ending up with misidentifications. So our concern now is the depletion of fish catch in inshore waters. Abundance of many commercial important fishes are declining. Fishes complain, size of fishes reduced. Shark populations show declining trend. Saw fishes once widespread are now critically endangered in an extinction phase. Invariably, an increase of the bycatch discharge in this region. So, Dr. Mahapatra also pointed out the same things. Both fresh and marine waters appear to be declining globally. Marine fisheries primarily threatened by overexploitation, pollution, and habitat loss. All ecosystems are vulnerable to the impact of climate change. The freshwater fishes, they are facing shrinkage of water bodies by reclaiming, deforestation leading to siltation of wetlands, gaming the river systems, habitat destruction, overexploitation and destructive fishing practices, introduction of invasive species, pollution of excessive use of pesticides, global warming and climate change. And marine fisheries are more commercial demand. These are threats. Target and non-target fisheries both continues to be there, highly susceptible to a variety of inshore demersal fisheries, including trawls and gillnets. They are simply screening the water, high level of stress pressure in the ecosystem habitat, threatened by overfishing, pollution, and habitat degradation, low fecundity, slow life history, but high mortality, so unable to recover the current level of exploitation. So how we can say these commercial important fishes, sharks, turtles need some sort of conservation measures. For this, awareness among fishers and fishing community is highly essential. Most of the fishes are not protected by existing law, and anticipatory laws are required. Regulations and actions to curb illegal, unreported, and unregulated fishing is required. Protected areas help in recovering the fish population to some extent. So proper identity of fishes, their extent of distribution and biology to help the managers to bring conservation strategies. And the first requirement is the taxonomic studies, and for this we require manpower. If some people are coming forward and working on the field of taxonomy to study the fishes, it will be more useful for the fish conservation and future of fishery studies. Thank you. Hello.
Okay, let's thank Mr. S.S. Mishra, scientist. So we can accept hardly one question if you have, because we are very much late. So hardly one question we can accept. Rest, if you have any questions, we'll provide the email ID. You can directly contact to Mr. S.S. Mishra.
I think we can invite Dr. Prashant.
Okay, sir. Okay, then. Thank you. Thank you, Mr. Mishra. Let's now, it's my pleasure to introduce Dr. Prashant Chakraborty. He is a professor as well as curator of Ecology, ACE Division, Louisiana State University, and he also have discovered many new species of fishes, particularly marine crabs, and also he have many publications in many reputed journals. He is also editor and associate editors of many reputed journals. So let's welcome Dr. Prashant Chakravarti for his lecture, and he will share, I think, most interesting part of this seminar. So I will request Dr. Prashant Chakravarti.
Hi, everyone. So I've been enjoying the last two presentations. Those were very informative. Let me share my screen. I hope I can do that. So Dr. Mishra, I think you're still in the presenter mode, but let me see if I can overtake that. I think Dr. Mishra, I think you have to turn off your screen sharing before I'm allowed to do so. Can you hear me okay?
Yeah, you are audible.
I'm able to? Let me see. It says share now. You must grant permission in order to screen share. Let's see. Sorry about the technical difficulties here. Yeah, it's not letting me. Let me see if I can. This will let you take over. Yeah. Can't share your screen. You must grant permission in order to share screen. Is it possible for Dr. Mishra to turn off his, or?
Dr. Mishra,
Let-
kindly
let
stop
me
presenting
try.
your screen, sir.
If the maybe Deepanjan can flip that. Okay.
I hope it's
Now,
aligned.
yeah, it should be fine now. Let's see. Can't share screen. You must grant permissions. Try window.
I hope the organizer has to permit, as you are logging in from outside India, that might be there.
Okay. I'm not sure the best way to do that. Yeah, it sounds like I need a permission to share the screen. Not sure what to do.
The organizer, please.
Sir, one thing, sir. Actually, there is no any option to allow, sir. Here, anyone can present the screen, and if problem is arising, then if sir will send his PPT, then I will try to present it, sir.
We can do that. So let me send you a link to my PowerPoint. I'm not sure exactly where to.
So kindly send me, sir. Let me present, sir.
Sorry about the. Never had that happen before. Let's see. Yeah, it's so big that I have to put it in Dropbox, so it might be a second here. Sorry, folks. Can I put it in the? How do I send it to you? Can I send it to Deepanjan?
Hello,
Yeah, I'm sending a Deepanjan.
sir. I am present there. Hello.
Yeah, I'm gonna send you my link to my presentation, so maybe you can send it. You can.
Okay. You send me an email. Okay. Thank you.
I just sent it. It's a Dropbox link. Apologies for that. Yeah, I've never had the permission voided there.
No, here also not asking any permission. When we are presenting, it's not asking for any permission. But
Yeah, it's
দেব আছে ওখানে? হ্যালো,
আছি স্যার, যা যা যা, I'm present স্যার।
দেব কি আছো?
send it to you on your
হ্যাঁ স্যার, আছি স্যার, আছি স্যার।
হ্যালো,
হ্যাঁ স্যার, আছি।
ওনার ব্যাপারটা একটু দেখুন তো।
স্যার,
ওইটা বলছি ওনার মানে একটু দেখুন।
অ্যাকচুয়ালি এখানে তো কোনো পারমিশন দেওয়ার তো কোনো অপশন নেই স্যার এখানে, বুঝতে পারলেন স্যার? আমি দেখুন, আমি প্রেজেন্ট করলে কিন্তু আমি প্রেজেন্ট করতে পারছি। এই যে স্যার দেখুন, আমার স্ক্রিনটা হয়তো দেখতে পাচ্ছেন স্যার।
আচ্ছা আচ্ছা আচ্ছা।
ওকে স্যার, স্যার কাইন্ডলি সেন্ড মি
I was able to
পিপিটি স্যার।
I sent it to Deepanjan. I don't have your email.
Okay, 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 okay. Sir Dipanjan sir, kindly you present sir, then if possible, or otherwise send me sir.
I guess I can send it in the chat. Did I put in the chat? It's a Dropbox link. I just put in the chat function, the Dropbox link if you want to put it in there.
So please wait for a few minutes sir. Please wait for a few minutes sir.
Sure. Well, I'll explain a little bit about my background if that helps while we wait. Let me get my presentation up. So I'm currently in Ottawa, Canada, where I'm doing a Fulbright. So I'm usually in Baton Rouge, Louisiana, where I'm a professor in evolutionary biology and a curator of fishes. And you know, as the previous speakers discussed, you know, having collections in which to compare specimens is really important, and most species are going to be described with comparisons in museums. And so you may go in the field and find something that you're not able to find in a guidebook, or that doesn't quite match. And what you do then is hopefully have some comparative material. And so that's why museums are so important, so that you can match them up with what you have. Let's see. Were you able to get my PowerPoint up, or?
it's a big file. It's taking some time.
It is a big file. Sorry about that. I thought I'd be able to show it here.
Yeah.
Let's grant permissions. I've never had the permissions thing. Let me see if I can.
हाँ Dipanjan, हाँ बोलो. Professor, I have downloaded that myself, Dr. Mahavidyalaya. I am presenting on behalf of you here from here.
Oh, that's great. Yeah. So if you can share your screen, thank you for doing that. And I'll just say switch screen when it's time to do so.
I hope it's fine now.
It's fine. Yeah, great. So I'll just say next slide, and you can go ahead to the next slide. So again, I'm in Ottawa, Canada now. I hope. So this is me in my natural habitat. So as a fish curator, we've redesigned our collections a few times, and we have about 350,000 specimens currently, and we have one of the largest collections of vertebrates in the world, including vertebrate DNA. So we keep small little samples of the tissues of the specimens that we have here in order for them to be sequenced. And we send thousands of loans out a year in order to help people do that. And these are, this is me in the stacks. We have about 50 rows like this in different parts of the museum. And it's representing about 50 different countries, the fishes that we have here. Can we have the next slide, please? So these are some of the collections-based work that I've done. So my lab consists of postdocs and graduate students, and they go to 2 or 3 countries a year as well. Are people saying they can't hear me? I should allow them to see me though. So, so I was lucky enough to sneak into Thailand this year in January and February, so I'll talk a little bit about that. But the reason that we go to all these places is to conduct natural history research around the world in order to have comparative material so that when we do describe a new species from, let's say, from Mexico, we can compare it with its closest relatives, which may be in Australia. This is obviously a privilege to be able to do this, but it's the importance of museums. Can I have the next slide, please? So this is me in my other natural habitat. So this is what we do in the field. I don't know if, how many of you have had the experience of either collecting on a ship, on a cruise in the oceans, or seining in fresh waters. This is actually in Tanzania, just a couple years ago, and we're collecting gobies and other creatures in this mangrove habitat where the water has gone up and down due to the tides over 9 feet. So what we're able to do here is collect these specimens. It was actually a research being done on mangroves and their importance, and we were collecting in order to voucher specimens. So vouchering is where you take a specimen and preserve it so people can understand what we have in the future, and to prove the identity of the species that you have. As you heard from the previous speakers, that there's often difficulty in establishing the identity, the actual taxonomic identification. Now, having traveled to a few places, I've been able to describe a number of species. These are the new species of fishes that I described. So there's some deep sea forms like the batfish above, and then there's a number of cave fishes. So a lot of my work is in caves these days, and some marine fishes too. So actually, if you can hit the next slide or have the, yes. So what's not as, no, sorry, the previous slide with the names up there. So new species are wonderful to find, but another part of taxonomy is the revision. So I've named a number of new genera and revisions of genera. One of the ones I'm proudest about, and under new genera, the first one is Carla, which is actually the ponyfish genus that I named after visiting Sri Lanka. So those ponyfishes are the ones on the bottom. You may call them flipmouths or have another name for them. But these fishes are marine fishes that were pretty poorly known until pretty recently. Myself and some Japanese researchers have been working on them for some time. Unfortunately, I've been to India, but I have not been able to collect in India, and you know, the complications with the Biodiversity Act have prohibited many foreign researchers from collecting in the same way that I would be able to collect from, say, Bangladesh, and we can talk about that later. Let's go to the next slide. So names are important. So this is not a phylogeny of fishes. This is actually a phylogeny of COVID-19, the virus that causes coronavirus. So names matter. As you can see early on in COVID-19, the names were very different in how we established the publication record. So names matter. Can we go to the next slide? So part of obtaining those names is figuring out who's related to whom. And so what we're seeing here is the world and how it's spread. So we're talking about fishes, but we're using the same technology to understand even something like how coronavirus is spreading around the world. And so, for instance, from this phylogeny, we were able to better understand that the coronavirus, the COVID-19 in New York City, was mainly from European stock and not from Asian, right? So even though it may have started in China, in Wuhan, the virus spread and mutated in different ways. So that's something that can be articulated better when you have the proper names and a proper phylogeny. And systematics is both phylogenetics, building trees like this, and taxonomy, naming and organizing. Can you have the next slide, please? So I obviously don't do that with viruses. We do that with fishes. This is a phylogeny of cichlid fishes that I worked on. The fossils on the right, the bottom one is a new one that I redescribed from Haiti. And it was previously in the genus Cyclosoma, and we put it in the genus Nandopsis based on its morphological features, particularly its teeth. So why is that important? Why do we need to change the names? Well, by changing the position of the fossil on the phylogeny, we were able to say that the cichlids in Haiti, where this fish is from, have been there for at least 10 million years, which is the age of the fossil. And so you're able to shift the age of the fossil and the age of the phylogeny in order to better understand how old the family is. So even a few taxonomic changes can have big repercussions for how old or how the tree is dated overall. So if we can go to the next slide. So in a phylogeny of gobies, so these are cave gobies. There's one from Australia that we described and a number from Madagascar that we described. If you can click through these, you can see, so what the phylogeny taught us, Dr. O'Neill, if you can move the slides. So what it taught us is that because of the name changes and the redescriptions and the taxonomic information, that these fishes were last together when the eastern part of Gondwana, the southern continents, were together. So if you want to click through this, Dr. O'Neill, if you can click the next few slides. So you can see India is part of that, and obviously there's a big gap. But these fishes, which are freshwater fishes in caves, didn't cross the Indian Ocean. So the taxonomy, the systematics, the work that we did helped establish the relationships here. This is another cavefish that we were working on in Thailand. So this is a CT scan, and you were able to see the pelvic girdle connect to the vertebral column. So that's a hip. You have hips. Fishes don't have hips, except for this cave angelfish from Thailand. So CT scanning, after we were able to see what this fish can do, we were able to scan the fish. And this is part of taxonomy too, right? Is understanding the anatomical features. And now we have the ability to look at these with CT and MRI scans. If you can click the next slide. So this particular fish, this is the cave angel, the Cryptotora from Thailand that we observed this year. So you can see it coming out of the water, walking on land. Most fish can't do this. They climb waterfalls, so this is a waterfall climbing fish. It's pretty remarkable. And again, it could not have done this without having this connection, which we were able to see over CT scanning. So this is not a mud skipper. This is a blind cavefish that has pretty remarkable powers to swim upstream. So if you click on the next slide. We're still looking at the anatomy here, but if you look at the fish in the aquarium, it actually lifts its head. And fishes don't have necks, so how it's able to do that anatomically, it's pretty interesting. And how cave fishes become cave fishes. We know this is a member of the riverine loaches, the Balatoridae, and so taxonomy may sound like an old science, and it is an old science, but it has many modern techniques as well. So if we can click the next slide. So I mentioned CT scanning. This is actually MRI work, so magnetic resonance. So these are pony fishes again. So these are bioluminescent fishes. And you can see we've plated the bacteria that the fish co-opt in order to be bioluminescent, and they glow, right? So the fishes glow underwater. They flash like some flashing insects do, except beneath the sea. You can see us plating up the Vibrio cholera, or it's not cholera, another Vibrio that allows them to luminesce. And on the right are the MRI scans, so that we were able to observe the light organ, which is a donut-shaped organ around the esophagus of the fishes. And when you dissect them, it's actually very hard to see the light organ because it collapses and it's very mushy. So when you do an MRI, you can see in situ what these look like. And you can see the green, there is the light organ, the fluorescent green, and they're in different shapes. And the pony fishes don't look very different. The previous speaker, Dr. Mishra, was explaining how Secutor was in a different genus. One of the genera of pony fishes is Secutor, another is Lagnathus. And we actually use the light organ shape to define the different genera of pony fishes. So you can use very modern techniques for taxonomy. And because we were aided by MRI studies, we were able to better establish that taxonomy. Can you, next slide, please, Dr. Mopton. So pony fish is it's too bad the volume's not on. So these fishes also communicate with sound. So underneath you can see the audio file where we graphed the sound. They have different ecologies too. So I mentioned you can do a lot of work in a museum, but it's also good to go out and observe these animals in life. You can learn a lot about the behavior, the ecology of these animals, which can also be part of the taxonomic features that you use to define these organisms, right? So taxonomy is defining what you observe in order to organize the tree of life. So let's continue, but these guys make neat sounds. This is an example of a study from my lab group. So Bill Lute, who is a PhD student in my lab, I would consider this a sort of modern taxonomy. Not that there's any need for people to do this kind of work, but we'll see more and more studies like this. So taxonomy is not just descriptions of species, but also synonymies. And so what Bill and I did in this paper is we synonymized these two surgeonfishes on the left. So the two Prionurus, which are these big, ugly surgeonfish. They were found from the west coast of the Americas, including the Galapagos. And using genomic work, so we looked at thousands of genetic markers, ecological work, comparing their diets, comparing their habitats, and morphological work, traditional morphological work, including measurements. We were able to establish that these were exactly the same species. So there's no taxonomic differences that lead to these being the same, the different species. They're the same species. And so they were synonymized with the senior synonym. And so this is an example of a modern taxonomic study. I think this is in molecular ecology, if you want to look this one up. Next slide, please. So a lot of my work, the previous speakers have mentioned barcoding genes, which is CO1 or sometimes 16S. We use, with a similar techniques, you can get not just one or two genes, you can get hundreds and sometimes thousands of genes. So there are particular genetic markers that you have, that fishes have, called ultra-conserved elements. And these markers are almost exactly the same across all vertebrates. But adjacent to those core regions, the core UCEs, are very highly variable regions that allow us to determine relationships through traditional phylogenetic studies. So this is a very neat kind of and valuable tool that is, may sound complicated or expensive. And I won't say it's not complicated and expensive, but in 10 years, this will be even cheaper. So just like barcoding genes were perhaps cost prohibitive 10, 15 years ago, that's something that more people can do now. So let's go to the next slide. So we've used these markers to figure out the relationships of some major taxonomic issues within fishes. So particularly the carassiforms, the tetras, have been split into two families. One is more closely related to the catfishes, the other to the knife fishes. If you continue through these next slides, we looked at the electric fishes and established their relationship actually fits with the electric signaling pattern that they have. The reason that the phylogeny here is blurry is because each line represents one of 1,000 genetic markers, right? And so you're looking at how the relationships can find a cohesive explanation for the relationships of these fishes. The electric fishes, the gymnotiforms here, are the neotropical knife fishes, which include the electric eel. Next slide, please. Oh, next slide after this. So this is, so I talked about ultra-conserved elements, UCEs. We also used exons for this relationships of gobies. So gobies are a mainly marine group that is found worldwide, and their relationships are very difficult to untangle. And we used exons, which are protein coding genes. And we used 1,000 protein coding genes to figure out the relationships here. I will say the more data you have, the more problems you will have. So one of the things that I've learned is that, you know, the paper ends up being more about the data and processing and how you figure out the relationships more than the fishes themselves. And for me, although that's unfortunate, what these data can be used by other researchers and by myself to better understand the taxonomy behind here. So we named families based on these new phylogenies, and that's part of taxonomy as well. Next slide. So this method that I've been talking about for ultra-conserved elements and for exons is called target capture. And so in the past, if you wanted a barcoding gene, you would extract the entire genome of the animal, of the fish, then you would sequence just a portion of that genome with one set of primers. And instead now you can throw a whole bunch of probes onto the genomes and amplify those if they're similar. And you can do that with exons and with ultra-conserved elements pretty easily and relatively cheaply relative to how much that would cost with traditional techniques. Next slide. Right, so this is called genome reduction because of the amount. So if you can click through these three, it's okay, you can go through the next 3 slides. So we've talked about ultra-conserved elements as target capture. Next slide. Next slide. Yeah. So there's exon capture, which is looking for protein coding genes, which can tell you about a little bit more of the function of each of those protein coding genes. Next slides. Next. You may also encounter people using RADseq, which is a more cost-effective way if you want to look at a population level. My students have used these for distinguishing groups across rivers, and this technique is getting more and more robust as well. Next slides. So the problem, as you may know that people are now looking at genomes. We've sequenced a few genomes. There's still less than 500 fish genomes available, but those genomes are problematic because let's say the lungfish, the African lungfish or the Australian lungfish or South American, have some of the largest genomes known, and they're 100 times bigger than our human genomes, so they're enormous. And then the smallest vertebrate genome is also a fish genome, the fugu, the second genome done after humans, and its genome is so small for the pufferfish that it's smaller than many invertebrates. So how do you align something that's 100 times bigger than something that's 100 times smaller? So those are issues that will come as we enter the genomic era. Next slide, please. Yeah, so you'll have to filter the data in some method. So the genome sizes, again, vary widely from the giant lungfishes to the tiny fugu. Next slide. Right, and so synteny is how you align those different portions of different size genomes. And if you're looking for a research area that relates to genetics, synteny is a big part of that. And so it's a problem that everybody will have to dealing with systematics, at least in the future. Next slide. So I don't want to discourage anybody. That's sort of the genetic aspect of it, but I'm a museum scientist, I'm a curator. I do a lot of molecular work, but I'm also a traditional taxonomist. So let's go to the next slide. What will be the role of museums? So part of that, if you're working with specimens, keeping the data with the specimen somewhere. So we're all on the goal of building a complete tree of life and understanding who's related to whom, but it'll just be a tree of names unless we add the data to it. So those data layers of the ecology, of the molecular aspects, of the taxonomy, of where the specimens actually are, is part of the curation that we need to do. So as biologists who work with specimens, it should be our prerogative to find how to connect all these data together. And as the previous speakers mentioned, many specimens don't have vouchers, don't have a linked specimen where you can check the identification. So the tree of life won't be worth as much if the data isn't connected. Next slide. So the genetic sequences here, as the previous speakers talked about, if you use barcoding like CO1, I created this nomenclature called GeneSeq, GENSEQ, where you can actually see if people link the voucher to the genetic sequences. So most of the genetic sequences on GenBank don't have any voucher listed with it, and that's a problem. And the more type specimens that are listed, the better. But most people don't even have a photo voucher with their genetic sequences, and we're working with GenBank to change that. Next slide. So we need to add that structure back onto the tree of life, whether it's ecological or distributional data. Next slide. If we add the fossil data, then we'll be able to figure out over time how things have changed. Next slide. So with the paleo data, we'll be able to unzip the tree of life and figure out when things happen and how things have changed. Next slide. But as I mentioned, there's a lot of issues with data. The more genetic data you have, the more computationally intensive it is. Most of the individual genes will not tell you the tree of life or the actual relationships. So you have to figure out how it's misleading you. So next slide. So it's similar to actually how your own genome, because it's a mix of your parents and then a quarter of those ancestors, how that is mixed is a similar problem that we see over the time scale of millions of years. Next slide. So I've had my genome sequenced. I've added the people here that I imagine I would be related to. This is a joke, obviously, but despite my accent, I do have Central Asian roots, as you can tell from my name. So, but the relationships that you see here, the point of this slide is that my brother may have a very different genetic test results. Maybe not very, very different, but different enough. And the same thing would happen between two catfishes in the same river in India, right? So it's much more complicated when we start explaining the genetic roots of organisms, even humans. And we're still figuring out, we're in the early days of genomics. Next slide. So there's a goal to sequence all the eukaryotes, all the multicellular life on Earth in the next 10 years. They won't be able to do it. Next slide, just to show this graph. So very few of them have specimens connected to them. And we don't know what genome sequence is attached to without having a voucher. And that's a point I'm really trying to establish with genetic researchers. Next slide. So for instance, the genome of Mola mola was sequenced pretty recently. And it's one of the most conspicuous animals on Earth. It's an elephant-sized sunfish with a brain the size of a peanut. It's a wonderful animal. The genome was very useful to have, but Mola mola was split into 3 species. And the genome was taken from a blood sample from a specimen in Florida. So whose genome is it? So taxonomy still matters. We may be in the age of genomics, but the taxonomic principles of keeping vouchers and comparing specimens is key even into this modern science. And a genome like this is basically descriptive work. So you're describing the complete DNA of an organism in the same way that you might count fin rays or count different the vertebral column, etc. So genomic work currently is very much descriptive. Next slide. People outside of taxonomy are now establishing what we call homology. So they're looking at comparisons of fins and limbs. And so there's all new techniques like CRISPR-Cas9, which permit you to compare the genetic results or change the genes of one organism to see how it affects the phenotype, the body. And this is not yet part of taxonomy, but part of taxonomy where we can perhaps collaborate with geneticists. Next slide. So this may be discouraging if you don't have access to these tools. Not very few people have access to those tools. But I like this quote from Ed Wilson to remind me of the real reason that we do this work. So don't let anyone tell you that taxonomy and simple exploration, boots on the ground, is finished. It's not. We still need people on the ground discovering and describing new species, particularly in places like India. Next slide. So I'll go through these pretty quickly. It's okay if you skip these. So we were looking at, our big blue planet, but there's really just one ocean, right? We might call it the Indian Ocean, we might call it the Pacific, but they're all connected in one giant ocean. This is maybe a different view of the oceans than you've seen before. And so how do we get, 15,000 freshwater species and 20,000 marine species without barriers here? A third of marine species are connected with coral reefs. So how is speciation happening in the oceans? Next slide. This is one of the most remarkable views of Earth I've ever seen, because the big ball over the Midwest of the US is all the world's water. So all the oceans fit in a ball that's the distance between Mumbai and Kolkata, right? So it's 860 miles in diameter. It's about 1,200 kilometers or so. Sorry, I'm not so good at the conversion. But that ball is all the world's water. And it's all the oceans, all the water that's in you, it's all the water in the rivers and lakes is in that big ball. So it's not as much water as you would think on Earth. The smaller ball next to it is actually all the world's fresh water. Most of the world's fresh water, 90% of it is ice, right? And then the tiny ball next to it is all the world's flowing fresh water. So we have remarkably little water, and so it's an important research to remember to keep valued. And half of all fishes are found in that tiny ball, and the other half in most of that larger ball in terms of species number. Next slide. And you can see the rivers here and lakes, which look like they're spread all over the world, but again, look at the total amount of water is very minuscule relative to the marine realm. Next slide. And just a reminder that we are still need more people to describe the planet's species before they go extinct. So less than 10% of the world's species are described. There's about 35,000 species of fishes described. There's probably closer to 60,000. So many species yet to be described. I bet many of them are from India and China. And so we need more taxonomists, more boots on the ground. Next slide. So just a plug, and excuse the plug, I have a book coming out next year, A Brief History of Life Through Evolution, which is going to be published with Penguin Random House India. And next slide. I also have a more traditional science book that's coming out, Methods of Fish Biology. Some of you may have seen the first edition, which came out 30 years ago. This won't come out in 2020, but 2021. If you're curious about more methods and taxonomy, this is an edited volume. So just look out for those next year. Next slide. So just in conclusion, the future of fish taxonomy will include new tools like CT scanning and genomics, and old tools. We still need people to do measurements and X-rays, etc. Old tools that I still use to describe new species. I'm describing a new species from Colombia right now, and we're using traditional methods because we don't have access to the specimens, most of the specimens. There are many species left to discover, and collaboration is key. I hope in this new COVID era that we're learning better to collaborate across the world. Next slide. So just to thank all of you for watching. I know you're supposed to end at 8 p.m. your time, which is just about now. It's 10:30 in the morning here on a Sunday. So I want to be cognizant of that and thank the organizers of the symposium. Thank you.
Okay, let's thank Professor Dr. Prashant Chakraborty, curator, and also is the professor of Louisiana State University, for giving us a very valuable time. So thanks. And now we are almost very late, so I'll request all the audience, if you have any questions, please let us know. We'll send it by email to Professor Chakraborty, and later on we'll acknowledge the answer. So let's thank to Dr. Professor Prashant Chakraborty for his valuable time. Thank you, sir. So now I'll request our coordinator of IQSC, Dr. Samanta, to give us vote of thanks. So, Dr. Samanta.
Thank you, Srinivanda. Respected teachers, respected resource persons, and my dear students, we have successfully completed our one-day international webinar on fixed taxonomy. So all of you are very cooperative, and that helped us to make this event a grand success. I am sure students of our college as well as students of other colleges have been benefited from this program. So students can explore this field further. This field is very rich, and large part of our country is surrounded by sea. So a wide varieties of species are easily accessible. So they can further explore or investigate this field. For further guidance, I would recommend them to contact Dr. Deepanjan Ray, who is the HOD, head of the Department of Zoology Department, Bajkul Milani Mahavidyalaya. So I do not want to elaborate my talk. So this is we are very near to the end of our program. Let me take this opportunity to thank the research persons for giving their valuable time. All of them are expert in their fields. So we are thankful. They have shared their knowledge and time with us, with our students. We are enriched by them. Also, I am thankful to all the faculties of Zoology Department, especially to Dr. Deepanjan Ray, who is incidentally the convener of this program, and Mr. Habibullah Malik, who is the secretary of this program. So they have organized this beautiful program. Students are very much interested, and they are asking lots of questions in the chat box. We cannot answer all of them. So, but we will try to answer them in email. Also, let me express my gratitude to Dr. Piyush Kanti Dandapat, who is the teacher in charge of our college, and also Mr. Ardhantu Maiti, MLA, who is the Honorable President of Governing Body. So with this, I end this international webinar. Thank you very much, and good night.